To watch these important lessons, subscribe to DP Education's YouTube channel right now. Click on the bell icon to stay updated on the latest lessons. Sri Lanka's largest free online school, DP Education.
Hello my dear students, welcome to today's lesson. So I am going to discuss with you all the questions given in your textbook related to the lesson inheritance. That is the 20th lesson of your grade 10 science textbook. So in the lesson, in the previous chapters, we discussed the lesson where we started off by discussing the characters inherited by the human population. So in that we discussed the common inherited characteristics. For example, the color of skin, that is the complexion, then nature of hair, curly or straight hair, then how the earlobes are fused or divided earlobes, then how the thumb is, whether it's bent or straight, then whether there is a dimple or not, and how the thumb is positioned when you cross your hands together. Like that, we discuss some common inherited characters. Then after that, we discuss the rare inherited characters like albinism, where there is no melanin pigment and the skin is white in color, a different type of white color, and also the existence of or the occurrence where the dactyls are fused together, fingers fused or the toes fused together, syndactyl, or additional occurrence of additional fingers or toes, that is polydactyl. Those were some of the rare inherited characters, and even the color of the pupil. Then thereafter we discussed how Gregor Mendel, father of genetics, carried out experiments using the garden pea plant, and he got many observations and tried to explain how characters are inherited, although he did not have the knowledge of DNA and genes. So based on that, we were able to write and explain how new generations, the F1 generation and F2 generations, were formed. Thereafter, we discussed what genes, DNA, then genotype, phenotype, homozygous chromosomes, homologous chromosomes, homozygous genes, and all those terms. Thereafter, we saw how sex is determined in man, and then the disorders due to sex-linked genes and gene mutations. And finally, we discussed how the gene technology, knowledge of inheritance, can be used as the recombinant DNA technology and how it is useful in different fields. So that was the lesson. And I'm sure you all have already answered the questions given in your textbook. If not, you all answer it by yourself first, and then you can listen to my discussion. So I'm going to start off with the exercise, the first question. So here you can see red-green color blindness is an inherited disease. Select the correct genotype of a carrier female. Now normally, you all know students, the red-green color blindness, it is caused by a recessive gene that is linked to the X chromosome. So when we say a carrier female, normally we get the chromosomes like this. Both are X chromosomes. And if it is a carrier, we normally use a capital C and a simple c. That is for a carrier female. But here, if you look at the answer, students, now here they have shown it as X0, X0, then X simple c, X0, that is the second answer. Third answer, X simple c, Y. Then fourth answer, X simple c, X simple c. They have only used the letter simple c. There is no capital C. Instead, they have used a 0. So, if there is this gene, recessive gene, that is responsible for the disease. Recessive gene, simple c, is responsible for the occurrence of color blindness. Now, if this recessive gene is not there, then there will be no disease. So that can be indicated in a different way. So instead of having capital C, simple C as the carrier, we can also show it this way. Now if these are the X chromosomes, we need to have a simple C to be a carrier, but the other gene, if it is not simple C, then it is a heterozygous condition. Now this is a heterozygous condition. If we had 0 and simple C, this is also a heterozygous condition. So this can be considered as the carrier. So X, 0, X, C can be a carrier. If both X have simple C, both simple C, homozygous recessive situation, then it is a disease condition. If there are no chromosomes, no recessive chromosomes, then of course the person will be healthy. Here of course we don't have to bother about the male because they have asked for the carrier female. So X and Y situation we don't need to consider. So let's look at the answers. Now X0, like I said, there are no recessive genes. So it's basically like having the dominant genes. So that is a healthy individual. So this is healthy female. Then here it's similar to this. X simple C and 0. So X0 and simple C or X simple C and 0. That is the correct answer. That could be the carrier female because one chromosome has the recessive gene, other one does not. So that is a carrier. Then if you look at this one, X simple C 1. This is a disease male. Anyway, we are not looking at males because the question is to identify the genotype of a carrier female. And here we have simple C, both in X. That means the female have the disease female. So then the answer has to be this. X simple C, X0. One has the recessive gene responsible for the color blindness. The other chromosome does not have that recessive gene. So that will be a female carrier. Is that clear to you all? So although you are used to this system where we use capital and simple letters for dominant and recessive genes and we show the chromosomes like this because it's a X-linked gene. But if they give the answers in this method also, you all have to look at the answer. You have to think, since you know the concept, you have to apply that knowledge. So here, simple C means the recessive condition. That means it's responsible for the occurrence of the disease. But if there is no recessive gene means, then you will not get that disease. So then based on that, you have to look at the combination. X and X combination and you need to find the answer. So for this question, red-green color blindness is an inherited disease. Select the correct genotype of a carrier female. So this is the genotype of a carrier female. X simple C, X0. I'm sure you all can understand that. So look at the question. What is the percentage of parental genotype? What are the parental genotypes? Capital B, the dominant homozygous genotype. Simple B, recessive homozygous genotype. So here, they want you to find the percentage of capital B and simple B obtained in the next generation. That is the first generation. So that is what is meant by progeny. Progeny means offspring. From the parents, the new generation that is formed is known as the progeny. So to do that, you are familiar with the ratio also, the Mendel's ratio. If you remember, F1, all are heterozygous. All have the dominant character. So if it is capital B and simple B, all the first generation will be only capital B, simple B, a heterozygous situation. If you want, you can write it and check. But if you remember, with that knowledge, you can answer it. So we have the parents, BB, then we have the gametes. It's only capital B, here simple B. So F1 generation, everything will be like that. Heterozygous condition. Genes. We do not have capital B or simple B. So there are no parental genotypes in the first generation. So what is the percentage? 0%. No phenotypes that are similar to the parents. That is homozygous dominant character, homozygous recessive character. Is that clear to you all? Because this is the heterozygous condition. So if we answer the question, here we have what is the percentage of parental genotypes received to the progeny if capital B is crossed with simple B cross is done. 100%? No, because 100% it's going to be capital B, simple B. 75%, 50%, 0%. So it has to be 0%. So here students, we have answered both the questions. First one, as you can see, the second answer. Then the second question, so here you can see what is the percentage of parental genotypes received to the progeny if capital B and simple B cross is done. So with the cross, when it says progeny, I showed you all how we get the first generation. So we saw that in the first generation, there is only heterozygous condition, capital B, simple B. There are no genotypes similar to the parents. That is why we took the percentage as 0. But in an instance, if you get a question where they refer to the F2 generation, or let's say they say the second progeny, they say refer to it as first progeny and second progeny, or F1 generation and F2 generation. If you take the F2 generation, then of course you all know the genotype and the phenotype. F2 generation, what is the genotype? The ratio will be 1:2:1. Phenotype will be 3:1. But in that 1:2:1, you get one homozygous dominant character and one homozygous recessive character. So if we write that down, here we have got the F1 generation. Now if you get the F2 generation, I am not doing the full explanation. I am just writing the ratio. F2 generation, genotype is going to be capital B is to capital B, simple B is to simple B. And this is going to be 1:2:1. So here, these two, this one and this one, both are similar to the parents. This is the dominant character, this is the recessive character. Both are homozygous and they are similar to the parents. So out of the 4, 2 of them resemble the parents. So then, what is the percentage there? If 2 out of 4 is equal to the parents, for 100, you will get the ratio as 50%. So that is if it is for the second generation. So when I answered the question, I considered only the first generation. You all have to keep this in mind as well. If they refer to the second generation, then of course it will be 50%. But if we look at the question, here, what is the percentage of parental genotypes received to the progeny if BB, that is capital B, is crossbred with simple B, cross is done. So there, since they say progeny, that refers to the F1 generation. That is why we have taken the answer as 0%. Is that clear to you all? So this is something you all have to remember, students. So with that, I will move on to the next slide. Third question. Parents with normal complexion got a child with white skin. Is it possible to happen? That's the first part. Second one, explain your answer with the knowledge of genetics. Now here, parents with normal complexion got a child with white skin. So you can take it as normal skin, the normal color, but the child became fair. That is also possible. Otherwise, when we say fair child with white skin, now you can remember albinism is also a condition where the white skin, that is an abnormal white color. So we can consider the child as having albinism also. Both are possible. How is that? Now you all can remember there are some characteristics that don't go from one generation to the other. They skip a generation. There is a possibility that there were people with white skin in the generation of the grandparents, but the parents did not have that characteristic. Again, the children got the same characteristic. So, if a characteristic skips a generation, then also you can get it in the next generation. Now, for example, we saw in the previous one, capital B. Simple B is not expressed in the first generation, but it is expressed in the second generation. Like that, the white skin may not be expressed in the first generation. They are the parents here, but it is expressed in the next generation. So, that is also possible. Otherwise, the gene that is responsible for the white skin, if it is recessive, then it could have been expressed due to the homozygous recessive condition in the children. So that is why the child has a white skin. So both are possible. You can give the explanation in either way. So to answer the question, first one, we can say yes, it is possible. You can just say yes, or you can say yes, it is possible. And then to answer the second question, explain your answer with the knowledge of genetics. So here we can say some characteristics skip one generation and then get expressed in the next generation. Therefore, the white skin was not expressed in the parent generation. But was expressed in the child. So you can answer it that way. Or else, so here we can explain it in another way also. The white skin is a character expressed by a homozygous recessive genotype. So this genotype did not occur in the parents, but they could have been carriers of the gene, and the child got both recessive genes. So if we want to show that, now let's say normal skin, I will take it as capital N. So white skin, we will take it as simple N, recessive character. So if both parents, carrier parents, capital N, simple N, capital N, simple N. So when you have the gametes, we will have capital N, simple N, capital N, simple N. And when that combines like that, so here capital N, here we have capital N, simple N, capital N and simple N, simple N, simple N. Now these are all normal skin, but here this one is white skin. So child who got the white skin. Although the parents had normal skin, they were carriers. So because of that, in the child, both the genes became recessive and the character was expressed and the child got white skin. Is that okay? So this is what we discussed before also. When there are marriages among blood relatives, there is a possibility the recessive genes are there. So because of that, high chances of the child getting that disorder. Here it doesn't say it's a disorder, but this is something similar to albinism. Albinism is caused by mutated genes. But still, you have to remember that. If it is a disease condition, more chances of getting that. So that is possible. The parents do not have white skin, but the child has white skin because of this. If the parents have heterozygous genes, then one child can have white skin. So that is this child. The other three, they will have normal skin, but again, one child with both dominant characters, so very healthy or very normal skin, but these two children, they have the heterozygous genes, but also the skin is going to be normal skin. Is that clear to you all? So you can explain it this way. So here the question is, parents with normal complexion got a child with white skin. Is it possible to happen? Yes. And explain your answer with the knowledge of genetics. So you can either say some characteristics skip one generation and then get expressed in the next generation. Therefore, the white skin was not expressed in the parent generation but was expressed in the child. That is one way of explaining it. Otherwise, you can go into more deeper knowledge of genetics. The white skin is a character expressed by a homozygous recessive genotype. This genotype did not occur in the parents, but they could have been carriers of the gene. And the child got both recessive genes. So this is how you can show that. Normal skin capital N, white skin simple N because it's the recessive gene. So carrier parent means capital N simple N, capital N simple N. When the gametes are formed, NN, capital N and simple N in both parents. And when they combine, you get this 3:1 phenotype ratio, the Mendel ratio. That is caused by the recessive character. Recessive character is only one. Homozygous recessive condition where the child gets the white skin. Is that clear to your student? So I'm sure you all have also written a similar answer to this question. With that, I will move on to the next question. Fourth one, an improved organism by recombinant DNA technology has. So here, of course, students, the MCQ type questions and long questions are mixed together. So this is the fourth one, an improved organism by recombinant DNA technology. What do? You take a DNA from one organism, insert into another organism, or you can remove a part of the chromosome from an organism. That is also possible. So changed its genotype only. Gene combination is changed, that is the genome is changed. Because of that, the character expressed by the gene is also changed. So change its genotype only, wrong. Change its phenotype only, that is also wrong. Change its genotype and phenotype both. That is correct, because the particular organism is going to express that character. For example, let's say crop resistant plant. The character crop resistant plant is expressed by that genotype. So that is the phenotype. So that is possible by this. Or let's say phenotype where you have a larger fruit. So the phenotype will be changed. So change its genotype and phenotype both. No effect on genotype and phenotype both. That is also wrong. So the correct answer is going to be changed its genotype and phenotype both. Is that clear to you? So with that, we'll move on to the next slide. Question number 5. If you are given with a homozygous yellow seed plant where the green color, capital G, is dominant and yellow color, simple G, is recessive. So that is the information that you know. Now green color is dominant. So that is why we have capital G. And yellow color is recessive. So we use the letter simple G. That is the information given to us. How do you find out the genotype of the green colored seed plant? Explain your answer. So since green is dominant, it can be either homozygous, that is capital G, or heterozygous, capital G, simple G. So green color dominant can be either this or this particular genotype. Both genotypes give rise to the same phenotype, green color. But since yellow is recessive, then it is only in the homozygous recessive condition, that is simple G condition, that the yellow color will be expressed. So how do we identify? The question is to identify whether the green colored seed plant has the genotype capital G or capital G, simple G, whether it is homozygous or heterozygous. That is what we need to identify. So what can we do? You all know the way that the generations are expressed or generations occurrence of the F1, F2 generation. So what we can do is, we can theoretically write down the ratio, find out the ratio, will explain how the generation is formed. You have the parents, then you have the gametes, then the F1 generation. So you will get a ratio. Always we use the recessive yellow color, that is homozygous yellow color, that is the recessive combination. That as one plant, one parent plant, and the other one, for one instance, we will consider the homozygous dominant character. Then we will consider the heterozygous but the green color. So in both, the ratio that we obtain for the first generation will obviously vary, because from your experience, you all know that when we have different types of parents, the generation will be different. So at the same time, what we need to do is, experimentally also, we need to crossbreed the plants. So you take the yellow color seed plant and the green color seed plant with homozygous genes, crossbreed them, get the generation, look at the observation, get the phenotype ratio. At the same time, you write the way the generation will occur, get the genotype ratio and the phenotype ratio. Thereafter, we repeat the same procedure theoretically with the second type, that is the heterozygous nature. So then also you will get another ratio. So first I will write the theory part, then I'll write the practical, what we need to do the experiment, and then how we can come to the conclusion. So here, explain how the F1 generation will occur with both homozygous green seed plant and heterozygous green seed plant. So first we will write with homozygous green seed plant. This, of course, students, from experience, you all know the ratio. That is the normal ratio as the Mendel's ratio. All the first generation have to be of the dominant character. All the plants will have the green seed. So that is something you all have to understand. You can remember that. And the genotype also, it will be a heterozygous condition. So here they have said green colored seed plant. So then we have the P1 parent generation, P, that is capital G into simple G. So from that we will get the gametes. You will only have one type of gamete, capital G and simple G. Sometimes we don't circle the gamete. You can circle it also. That is fine. You can write it without circling it as well. So then you get the F1 generation. So F1 generation, when these two combine together, we will get capital G, simple G. All green seed plants. That is the observation you will get. Then the second one. I will use a different color. So second one with heterozygous green seed plant. So what will be the parent? Capital G, simple G and simple G. So we will have the gametes. Gametes are going to be capital G, simple G. Here it is simple G. Then, when they combine to form the F1 generation, these two, so this is how the combination will take place. So here, capital G and simple G. This capital G and simple G. Then simple G. So what will be the phenotype? Both of these will have green seed and these two will have yellow seed. So what is the phenotype ratio? So phenotype ratio, green is to yellow, 2 is to 2, so that will be 1 is to 1. You have 2 green seeds and 2 yellow seeds. So it has to be 1 is to 1. So then what do we do? This is the theoretical part that we have calculated. If the green color plant, green seed plant is homozygous, that is capital G, like this, then all the plants in the first generation should have green seed. But if the green color plant is a heterozygous plant, capital G, simple G, then of course you will get both green color seed and yellow color seed plants. And that also in the 1 is to 1 ratio. So then we have to do the experiment. So what do we write there? So I'll say experiment. Here you can say carry out a monozygote hybrid cross between the homozygous yellow seed plants and the green seed plants. So, how do we do that? Allow them to cross-pollinate. How do we make them to cross-pollinate? Deposit the pollens from the green seed plant on the stigma of the yellow seed plant and deposit the pollens from the yellow seed plant on the stigma of the green seed plant. You can explain that also. Deposit the pollens of green seed plant on the stigma of the yellow seed plant we can say. Also deposit the pollens of the yellow seed. That is the yellow seed plant on the stigma of the green seed plant, that is artificial pollination. Then what do? Then you have to get the seeds. One fertilization takes place, you get the seeds. So here, this is the first step. Then the second step, get the seeds of the plants. So then we get the seeds of the plant. So we are checking for the color of the seed now. So now that you got the seeds, that is the first generation. So this will be the first generation. This is the F1 generation. Now, if you were to look at the color of the flower or the shape of the flower or maybe the color of the fruit, you might have to color of the fruit, of course, you can observe it here. Color of the flower, you might have to plant the seeds again, grow them to observe the color of the flower. But here, of course, it is the seeds that we are concerned with. So then we don't have to carry out the rest of the steps. Now we have the first generation. You had the parents, you allow them to cross-pollinate by depositing the pollens of yellow seed plant on the stigma of the green seed plant and the pollens from the green seed plant on the stigma of the yellow seed plant. Then you allow them to produce the seeds and you look at the seeds. So those are the seeds that we need to observe. So once you observe the seeds, you will see a particular characteristics. So what are the possible outcomes? If all the seeds are green in color, then you know the green seed plant was the homozygous condition. But if you get the green seed and yellow seed in a 1:1 ratio, then you will know that the green seed plant, that was the green seed parent plant, had the heterozygous genes. That is how we identify the genotype of the green seed plant. Is that clear to you all? So theoretically you calculate, then you carry out the experiment, or you can do that simultaneously, because for the experiment you need time. But calculation theoretically you can be done easily. So you do the experiment, at the same time you have the calculation, explanation of how generation is formed, then you compare the ratio. Okay. So that is how you get the F1 generation. And then we write the conclusion. So conclusion. What do we do? Compare the color of seeds obtained with the ratio obtained. If all the seeds are green in color, then the green seed plant has the genotype capital G, capital G. If the seeds are green and yellow in the ratio of 1:1, then the green seed parent plant is heterozygous. That is capital G, simple G. That is how we conclude. So you compare the results obtained from the experiment with the ratio that you have obtained. If all the plants form green color seeds, then that means the parent green seed plant was in the homozygous state, that is the genotype is capital G, capital G. But if you get green color seeds and yellow color seeds in equal number, it may not be exactly equal. You all know from the data we have looked at before. When you get the ratio, when the number of plants increases, the number of seeds increases, the ratio becomes more accurate. So the closest whole number ratio will be 1:1. If you get that ratio, then the parent green seed plant will have the genotype capital G, simple G. That is how we conclude. So I'll again go back and quickly go through the procedure again. So here the question is this. If you are given with a homozygous yellow seed plant where the green color capital G is dominant and yellow color simple G is recessive, how do you find out the genotype of the green colored seed plant? So how do you find the genotype of the green colored seed plant? Green color is dominant, yellow color is recessive. So since green color is dominant, there are two possibilities of genotype. Gene expressions, it can be capital G or capital G, simple G, homozygous or heterozygous condition. So what we do is, we explain how the F1 generation will occur with both homozygous green seed plant and heterozygous green seed plant. So first, I have explained how the homozygous green seed plant will give rise to the F1 generation. So parents are going to be capital G and simple G, simple G. This is the green seed plant, this is the yellow seed plant. So gametes will be only one type, capital G and simple G. So the F1 generation all will have green seeds, green seed plant, all green seed plants. Then with heterozygous green seed plant, you carry out the same procedure. Parents, capital G, simple G, the heterozygous nature. Then you have the second parent, simple G, and the gametes. You have two types of gametes here. There is only one type, but you have to write it twice so that you get the correct ratio for the F1 generation. So F1 generation, you get two heterozygous green seed plants and two homozygous yellow seed plants. Then here, we have the phenotype ratio, green is to yellow, 2:2, that is 1:1 ratio. Then what we do is, we have to carry out the experiment. Carry out a monohybrid cross between the homozygous yellow seed plants and the green seed plants. So allow them to cross-pollinate. That is, by deposit the pollens of green seed plant on the stigma of the yellow seed plant. Also deposit the pollens of the yellow seed plant on the stigma of the green seed plant. So that is going to be artificial pollination. Then you get the seeds of the plant. You allow them to fertilize, then you will get the seeds. So once you get the seeds, you know what type of seeds they are. Normally, if you want to look at the color of the flower or something, you will have to grow them and then you will need to get the F1 generation. Then we have the conclusion here. Compare the color of seeds obtained with the ratio obtained. If all the seeds are green in color, then the green seed plant has the genotype capital G, capital G. If seeds are green and yellow in the ratio of 1:1, then the green seed parent plant is heterozygous, that is capital G, simple G. So that is how you identify the genotype of the plant. Is that clear to you all? So you have to show how the F1 generation occurs, and also you have to explain the experiment procedure. Then only your answer will be complete. I'm sure you all can understand that. So with that, students, I will move on to the next slide where we have the last question from your exercise. Question number 6. Sum of the genes in an organism is known as genome. So I have explained this to you all. Human genome has 23 pairs of chromosomes. In each chromosomes, there are a fixed number of genes responsible for various characteristics. That is, in autosomal chromosomes, the first 22 chromosomes. Then you have the 23rd pair, the sex chromosomes, that determines the sex of the person, as well as it has other genes responsible for other characteristics as well. For example, the gene that is responsible for clotting of blood. So you know, or the distinguishing between different colors. That is why the hemophilia disorder and the color blindness both are X-linked chromosomes. That is, recessive nature causes the disorder. So like that, there are other genes also in the 23rd pair, that is the sex chromosomes. So like that, we have different chromosomes having different genes in them. All that together, the sum of the genes in an organism is known as the genome. Under Human Genome Project, it has started to arrange the base sequence of genes. That is, they are mapping the genes of a person, humans. So they have the 23 pairs of chromosomes. They take each pair, say the first pair of chromosomes, and they identify the genes, and in that they identify the base pairs. Base pair sequence is the gene. So that is mapped so that they know the full correct order of the base pairs in the first pair of chromosomes. Like that, mapping is done for all the 23 pairs. So once the project is complete, they will know all the genes, all the base pairs present in all the genes of all the 23 pairs of chromosomes of man. So that is what we call as arrange the base sequence of genes. That is mapping of genes. That is known as the Human Genome Project, which is the statement that shows the harmful effect of the above to human. So related to this Human Genome Project, we can have certain advantages of X. At the same time, there can be disadvantages also. So they want you to identify the harmful effect. Harmful effect. So if you look at the answers given to us, use of gene technology as a remedy for genetic disorders. That is of course correct, no? Gene therapy. You replace the mutated genes by the good genes. So you don't get the disorders. You get the good characteristics. For example, if there is the gene that has thalassemia, you can replace it with the normal gene. So the person will not get thalassemia. So that is of course a good effect. Then the second one, ability to produce human with special features. That is something that will alter the people, the nature of people, characteristics of people. If you try to produce humans with special features, that will become a problem in society. So that is a harmful effect. Although we might think, okay, if we have special features, that would be good. But that will disturb the balance of the environment, as well as it can lead to so many social and ethical problems as well. So you can't have super people. You can have the normal people with the normal genes. But one thing, if there is remedy for genetic disorders, that is fine. But you cannot try to make the person better, having special features. That is not good. So this is a harmful effect. Then the third one, ability of the life insurance firms to identify the applicant's health conditions easily. So if you do a DNA test, if they have a genetic disorder, then the insurance firms, they can identify that. Like similar to forensic science, here also you can carry out the DNA test. You can identify the genes that can lead to disorders or diseases in a person, health conditions in a person. That is useful because there you are not going to change the person. The person is already there. You are just going to identify the problems, health issues related to that person. So that is also a useful effect. Then the fourth one, as a remedy for food problem using improved plants and animals by gene technology. Of course, that is correct. That is one of the main applications of gene technology, recombinant DNA technology or genetic engineering in the field of agriculture. So where you can solve the food problem by using gene technology. So that is also good for us. But the second one, ability to produce human with special features. This is like cloning or producing a different type of human being. Not the normal people who are there on earth. You try to modify them. That is harmful. So out of all these four answers, the second answer, ability to produce human with special features, is a harmful effect. So this is a harmful effect. So I'm sure you were able to answer this correctly. So with that, students, I have come to the end of this chapter where I have completed all the questions given in your textbook. So in the next chapter, I will discuss some extra questions with you.
To watch these important lessons, subscribe to DP Education's YouTube channel right now. Click on the bell icon to stay updated on the latest lessons. Sri Lanka's largest free online school, DP Education.